Hello students, welcome to Zoology Classroom. This is L. Mahesh, Assistant Professor of Zoology from Tara Government College, Sangareddy. In today's class, we are going to discuss about DNA structure. So DNA structure, as we all know, was discovered by James Watson and Francis Crick. For this discovery, they have won Nobel Prize also, and they have found that DNA is having a double helix shape. So DNA is very important molecule. So and what is the importance of DNA molecule? Let us see. So parts of DNA are acting as genes, and these genes they will produce proteins, and these proteins in turn they will control traits in our body. So all the characters that we see in our bodies and all the events that occur in our body, be it biochemical reactions, be it physiological activities, everything that happens in our body is controlled by one or other type of proteins. So without proteins, nothing happens inside our body. So each chemical reaction, each biochemical reaction, definitely it will be stimulated or catalyzed by one type of enzyme. At least one type of enzyme should be required for the occurring of any biochemical reaction. So, and we know that enzymes are nothing but protein. So, in this way, by controlling the production of proteins, by controlling the production of enzymes, genes, that means DNA, is controlling everything inside our body. So, all physical characters, all physiological things, all physiological activities, everything inside our body is controlled by indirectly DNA. So DNA is producing proteins, proteins are controlling our characters. So in this way, DNA is very important thing inside our body. In other words, we can say that it is having everything that happens inside our bodies. So right from starting from our birth to till the end of our death, everything will be controlled by DNA only. So it is very important for us to know the structure of DNA. As we have already seen, DNA will appear like a double helix in structure, and each DNA molecule, okay, each DNA molecule will be possessing two molecules inside it. These two molecules are called as polynucleotide chains. So two polynucleotide chains will be there. These two polynucleotide chains are attached with each other. They spirally coil around each other. Okay, they spirally coil around each other, and they are connected with steps-like structure, like in a ladder. So in this way, they appear like a ladder, and they are coiled around each other. So they also appear like a double helix. So each DNA molecule is having two polynucleotide chains. So each polynucleotide chain is made up of many nucleotides. So nucleotides are structural units of polynucleotide chain. So each nucleotide consists of three components. They are phosphate group, second one is pentose sugar, third one is nitrogenous base. Let us see in detail about these three components. First one is phosphate group. Phosphate group actually will be provided to the DNA by phosphoric acid. So chemical formula of phosphoric acid is H3PO4. So this H3PO4 provides DNA, this what phosphate group. So phosphate group will be taken into DNA from this H3PO4. And second component is pentose sugar. So two types of pentose sugars are there in nucleic acids. One is ribose. Ribose is present in RNA, and second one is deoxyribose. It is present in DNA. So only a small difference is there in between ribose and deoxyribose. Here we can see that this is second carbon in ribose. This is second carbon in deoxyribose. So the second carbon of ribose is having one hydroxyl group and one hydrogen molecule. And in deoxyribose, the second group is having two hydrogen molecules. So one oxygen is missing. So remaining everything is same both in ribose and deoxyribose. Only a small change is there in the second carbon molecules which are that are attached to the second carbon. So it is having hydroxyl group, it is having only deoxyribose is having only hydrogen molecule. So one oxygen is missing. That's why this molecule has got the name deoxyribose sugar. So one oxygen is missing in deoxyribose when compared to ribose. And third component of polynucleotide chain is nitrogenous bases. There are four nitrogenous bases which are present in DNA. One is adenine, another one is guanine, third one is thymine, fourth one is pyrimidine. Adenine, guanine, both these two things are called as purines. Thymine and cytosine are called as pyrimidines. So pyrimidines are having only a single ring-like structure. They are having hexagonal structure, whereas purines are having two rings. Out of these two rings, one is hexagonal ring, second one is pentagonal ring. So two rings are present in purines, one ring is present in pyrimidines. So pyrimidines, they are having more alphabets in this word, but it is having only one ring in the structure. Purines, they are having less alphabets in the word, but they are having two rings in the structure. So in this way, you can remember the number of rings which are present in purines and pyrimidines. And these are the purines and pyrimidines. So this pyrimidine, they are having only single molecules, okay? Cytosine, uracil, and thymine. These are the three pyrimidines, okay? So out of them, uracil is present only in RNA, it is absent in DNA. Thymine is present in DNA, it is absent in RNA. And purine structure, it is having two rings, one hexagonal ring, one pentagonal ring. So they are having two types of purines, adenine and guanine. Both these two are present in both in DNA and RNA. So now let us see how the nucleotide is formed. So this is green thing is pentose sugar. So pentose sugar, this is the first carbon. So pentose sugar is attached to nitrogenous base on its first carbon. So in each nucleotide, nitrogenous base will always be attached to first carbon of pentose sugar, be it ribose or be it deoxyribose, whatever it is, nitrogenous bases always will be attached to first carbon of pentose sugar. And first carbon, this is second carbon, this is third carbon, this is fourth carbon, fifth carbon is on the outside of this ring. So here, fifth carbon will be there. So phosphate group will be attached to the fifth carbon. So in each nucleotide, phosphate group will be attached to the fifth carbon of pentose sugar. Nitrogenous base will be attached to the first carbon of the pentose sugar. So this is a rule-like thing. So now four types of nucleotides are present in DNA. One is having adenine, another one is having thymine, the third one is having guanine, fourth one is having cytosine. So the nucleotide which is having adenine, is called as deoxy adenosine monophosphate. So we know that ATP, what is ATP? Adenosine triphosphate. ATP is adenosine triphosphate. So here adenosine is there. So adenosine and thymidine, guanosine, cytosine. Okay. So all these things are called as nucleosides. So how the nucleosides are formed? Here we are seeing nucleotides. Next thing is a nucleoside. When pentose sugar is attached to nitrogenous base, that means phosphate group is missing. When phosphate group is not there, both these two components are called as nucleoside. So adenosine is, when adenosine is attached to phosphate group, it is called deoxyadenosine. Okay, deoxyadenosine. When cytosine is attached to sugar, deoxyribose, then it is called as deoxycytosine. Deoxycytosine. When guanine is attached to deoxyribose, then it is called as deoxyguanosine. deoxyguanosine. And when thymine is attached to ribose sugar, then it will be called as thymidine, deoxythymidine, deoxythymidine. So deoxythymidine, deoxyguanosine, deoxycytosine, deoxyadenosine. These are the four types of nucleosides. When all these nucleosides are attached to one phosphate group, then they will form nucleotides. So what are the names of nucleotides? Deoxyadenosine monophosphate, DAMP, and deoxythymidine monophosphate, DTMP, deoxyguanosine monophosphate, DGMP, deoxycytosine monophosphate, DCMP. So these are the names of the nucleotides present in DNA. Whereas the nucleotides in RNA, they will be called as deoxy will be removed from them. So adenosine monophosphate, AMP, and uracil monophosphate, UMP. Thymine will not be there in RNA. Uracil monophosphate and guanosine monophosphate, GMP. Cytosine monophosphate, CMP, will be there in the RNA as nucleotide. So here we can say one thing that from ATP, nucleotides can be formed from ATP, from UTP, from GTP, from CTP. Nucleotides required for RNA can be formed. Why? Because when ATP loses two phosphate groups, it becomes AMP. So AMP is the nucleotide which is present in RNA. So in place of ribose sugar, ribose, if deoxyribose is added to AMP, then it becomes a nucleotide of DNA. So in this way, nucleotides can be formed from triphosphate molecules, which are having adenine or deoxyadenosine, thymine, guanine, and cytosine, like that. So here these nucleotides, okay, these nucleotides, they can form specific bondings in between them. So adenine, guanine, okay, adenine, guanine, thymine, cytosine were there. So but here adenine and thymine, AT, they can form bondings in between them. Likewise, cytosine and guanine also, they also can form bondings in between them. So both these two things, adenine, thymine, they form a base group. Cytosine and guanine, they form another base group. So two types of base groups are present in nucleotides when they are forming two polynucleotide chain. So here, adenine, it is the purine, thymine is pyrimidine. Okay, one adenine, one purine is forming bonding, base pair bonding with one pyrimidine. So adenine purine is forming a bonding with thymine pyrimidine. So here we can see that two hydrogen bondings are formed. This is one hydrogen bonding and this is second hydrogen bonding. So in between adenine and thymine, two hydrogen bondings are formed. And whereas guanine, this is second purine, cytosine is another pyrimidine. So in between guanine and cytosine, 1, 2, 3 hydrogen bonds can be formed. So three hydrogen bonds, they join both the cytosine and guanine. So in this way, we can see that the two polynucleotide chains will be formed. So this is one polynucleotide chain on the left side. This is another polynucleotide chain on the right side. So here, how the polynucleotide chain is formed, let us see that. So as we have already seen, so this is, let us consider this one. Okay, let us consider this lower nucleotide. So this nucleotide is having cytosine on its first carbon, on its fifth carbon, what it is having phosphate group. Okay, and this is second nucleotide from bottom. So the second nucleotide is having a guanine on its first carbon, phosphate on its fifth carbon. So here what is happening in between this first nucleotide and second nucleotide, the phosphate group of first nucleotide is forming a phosphodiester bond in between this third carbon of the second nucleotide chain. So this is first carbon, second carbon, and third carbon. So this phosphate group is attached to third carbon of the first nucleotide and fifth carbon of the first nucleotide and third carbon of the second nucleotide chain. So this is called phosphodiester bond. So this phosphate is attached with the both nucleotides with diester bonds. So this is ester bond, this is another ester bond. So two ester bonds are there. So that's why it is called diester bond. So phosphate group is attached to these two nucleotides with two ester bonds. So that's why it is called phosphodiester bond. So this phosphodiester bond is joining both these nucleotides. So dinucleotide is formed. In this way, the phosphate of second nucleotide attaches with the third nucleotide on its third carbon, forming the phosphodiester bond. So now trinucleotide. So likewise, fourth one also will be attached to the third phosphate group by phosphodiester bond. So tetranucleotide. So in this way, as the nucleotides keep on adding to this chain, the length of the chain will be increased. So finally, it will form a polynucleotide chain. So this polynucleotide chain is having a direction. So this is the first nucleotide. On the first nucleotide, we can see third carbon is having free end. Okay, third carbon is having free end. On the last nucleotide, we can see that fifth carbon or fifth carbon. Okay, fifth carbon will be having free ending. On the free end, it is attached to phosphate group. So in this way, this polynucleotide chain is having, okay, this polynucleotide chain is having 3 to 5 direction. So in this way, another polynucleotide chain also will be there. This also will be having 3 to 5 direction. So when both these two nucleotide chains come together, okay, so in these two polynucleotide chains, nitrogenous bases will form base pairs in between them. As we have already seen in the previous slide, thymine will form two hydrogen bonds with adenine, and guanine will form three hydrogen bonds with the cytosine. So in this way, by forming these hydrogen bondings, both these polynucleotide chains will come together and they spirally coil around each other. So here one polynucleotide chain is having 5 to 3 direction. The opposite polynucleotide chain will be having 3 to 5 direction, or vice versa, we can say. So this is the direction. So these two polynucleotide chains are anti-parallel to each other. This feature is called as anti-parallel feature. So both the polynucleotide chains are anti-parallel to each other. So here, this is the chain on the outside. It is having phosphodiester bonding linkages. So this is the backbone of the DNA. This is also backbone of the DNA. So these two polynucleotide chains on the outside, they are having phosphodiester bonding backbone. And on the middle, in the middle, we are having hydrogen bonding. So these hydrogen bondings are forming step-like structures in a ladder. And this is the backbone is very, very important. Backbone bondings are very, very strong bondings. Phosphodiester bondings are very, very strong bondings. Whereas these hydrogen bondings are very weak bondings. So hydrogen bondings can easily be separated, or they can easily can lose the bond again, can go, and they will allow both polynucleotide chains to get separate from each other. But whereas in a single polynucleotide chains, nucleotides cannot be separated from each other so easily. Why? Because phosphodiester bondings are very, very strong bondings. And coming to the some features of this double helical structure of DNA. So we have seen that this two polynucleotide chains are attached to each other with hydrogen bonding. So these hydrogen bondings are shown in between these two backbone structures. So here they are coiled around each other. Okay, they are spirally coiled around each other. So in this thing, what is happening? Each polynucleotide chain is taking a 300-degree turn. So let us imagine that this polynucleotide chain started from here, and it is coiling around the opposite polynucleotide chain. And when this chain comes to here, it is making a complete turn. So a complete turn. So okay, so this complete turn is taking 360 degrees angle. So when this polynucleotide chain is taking what? Complete turn. So in this complete turn, totally 10 nitrogenous base pairs will be there. 10 nitrogenous base pairs will be there. So if we take this one, the turn of this polynucleotide chain started here, and this turn, complete turn is ending here. So in between this complete turn, 34 nanometers difference or distance will be there. 34, here it is given 3.4, but it is not correct. 34 nanometers distance will be there in the starting of the turn and ending of the turn. So in each turn, 10 nitrogenous base pairs will be there. 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. So totally from here to here, 10 nitrogenous base pairs will be there. So, and in between each base pair, in between the successive base pairs, 3.4 nm gap will be there. So on the whole, 10 base pairs are there. So 10 into 3.4 makes 34 nanometers. So each turn is occupying 34 nanometers space, or a gap of 34 nanometers will be there in between the starting and ending point of the turn. And the width of these both these nucleotides, so starting from here to here, so 20 nanometers gap will be there. This is called diameter of the DNA. So diameter of DNA, 20 nanometers is the diameter of the DNA. So while these two polynucleotide chains are turning around each other, or piling around each other, they are forming two grooves. See here, we can see in this complete turn. So the gap in between these two turns is bigger than the gap in between these two things. So this is called minor groove. This is called major groove. So in each turn, okay, in each turn, two grooves will be there. One is major groove, another one is minor groove. Here also, if we consider this polynucleotide chain, so it started its turn here and the turn ends here. So in between this a complete turn, so this is minor groove and this is the major groove. So one major groove, one minor groove will be there. And these grooves, major and minor grooves, they provide place for the histone proteins and non-histone proteins to accommodate. So histone proteins, non-histone proteins will be accommodated in between these grooves. So while this DNA is forming nucleosomes and the condensation is started. When condensation of DNA is started to form chromosome, so the additional proteins, additional molecules will be adjusted in between these groove places only. So this is about the DNA. These are the characters of DNA molecule. So DNAs are, so far what we have discussed is bDNA or basic DNA. Most of the cells will be having this basic DNA only. But in some rare cases, we can see another type of DNA also, that is aDNA, and third one is zDNA. aDNA also will be similar to bDNA, but small differences will be there in terms of number of base pairs per turn. Like that, only small differences will be there between aDNA and bDNA. But zDNA is very completely different DNA from these two DNAs. So what are the differences among these three DNAs? Let us see. So aDNA is right-handed DNA, bDNA also right-handed DNA. So what is right-handed DNA? So here these turnings, so all these turnings are, they are turning at the right side. So right angle turnings will be there in this bDNA and aDNA. Whereas in the zDNA, the turnings of the polynucleotide chains will be having left hand. And diameter of the each turn, diameter of these DNA molecules, bDNA, as we have already seen, it is 20 angstrom diameter, and A form is having a 26 angstrom diameter, whereas zDNA is having 18 angstrom only. Base pairs per helical turn. So 10 base pairs will be there in bDNA. In aDNA, 11 base pairs will be there, and in zDNA, 12 base pairs will be there. So, and helix rise per base pair. Otherwise, the gap in between two successive base pairs is 3.4 angstroms in bDNA, 2.6 angstroms in aDNA, and in zDNA it is 3.7. So base tilt normal to the helix axis. Base tilt to the helix axis is 6 degrees in bDNA. It is 20 degrees in aDNA and 7 degrees in zDNA. So remaining two things are also not important. So it is enough for you to remember these 1, 2, 3, 4. These 5 things have 5. So these 5 points are enough to remember the differences among these three forms of DNA. So with this, today's topic is over. Let us meet with other class and other topic in next class.